right. Thanks, Mike. Today, we will talk about ADME-Tox profiling in drug discovery. In early drug discovery stage, the goal is to select the right candidate for further development. The assays in this stage are generally used for screen purpose, helping you to have a preliminary picture of your compound and to make an informed go and no-go decision. In this early stage, there are many compounds to be screened. Limited resources may prevent you from conducting detailed experiments on each of these compounds. We will often asked by our clients how to design a strategy so that more pieces of information can be obtained cost effectively. So today, Dr. Jackie John and I will give you an overview on how to choose right assays in early ADME-Tox screen. I will focus on bioavailability and drug interaction, and Jack will provide information on toxicology and safety pharmacology. ADME stands for absorption, distribution, metabolism, and excretion. So basically, ADME describes major steps of drug moving through the body. There are several important in vitro assays that will reveal your compound characteristics in each step. For instance, the permeability will related to absorption, and also solubility. It's also affecting the absorption, and then stability in cytochrome 50 or in UGT will affect the metabolism. So those are very important assays to have to run. And the result of this assay may be able to predict in vivo PK outcome if experiments are designed correctly, and help you to understand whether any changes in these ADME steps will alter important PK parameters such as AUC. These are the good ADME characteristics an ideal drug should have. On the other hand, if a compound does not have majority of these good features, it is more likely to have some problems down the road. The ADME screen assays are to identify these characteristics in the compound. Because of time limits in this webinar, I will only focus on in vitro permeability and metabolism studies. Okay, so first is permeability. Permeability describes compounds moving from one side of the membrane to the other side. Drug can pass through the membrane through paracellular passive diffusion, or transcellularly via passive diffusion, or transporter-mediated permeation. So here, the brown is uptake transporter, and the pink is efflux transporter. And sometimes, if a drug is a substrate of efflux transporter, it may have a difficulty in passing through the membrane from one side to the other side, depending on the location of the efflux transporter. So permeability may affect all steps of ADME, especially for those drugs use transporters as the main mechanism of passing through membrane. Permeability could be a rate-limiting factor of ADME. High oral absorption is a preferred characteristic of a drug. Permeability is one of the factors affecting the oral absorption. So our bioprint database show that the compound with apparent permeability coefficient greater than 5 have high possibility of good absorption. Permeability assays are tested using polarized cell lines such as KQ2 and MDCK. With KQ2 the most popular model, so it is gold standard, and MDCK used as an alternative method. To have a better understanding of permeability, the study should be conducted bidirectionally. That is from apical to basolateral, and also from a basolateral to apical. The parameters the parameter of permeability is apparent permeability coefficient. It is calculated based on Fick's first law. The common unit of this parameter is 10 to the power of -6 centimeter per second. If a compound has less than 10% amount of decrease in donor chamber, the permeability coefficient will be calculated using sink condition. How to interpret the results? And usually we, based on our bioprint data, we think that any compound has a Papp value less than 2 will have a low, it will be considered low permeable compound. Between 2 and 20 is a moderate, medium permeable compound. And if a Papp value greater than 20, it is a high permeable compound. For compounds with a low passive diffusion, transporters will play a more critical role. Transporters can carry compounds against their electrochemical potential gradients. And two types of transporters. One type is solute carrier transporters, and the other one is ATP binding cassette transporters. A majority of SLC transporters are uptake transporters, while all ABC transporters are efflux transporters. Various transporters are widely distributed in different eliminating organs and important target tissues. Whether a particular transporter is critical in ADME is often compound-specific. According to FDA and EMA, based on current clinical study data, the transporter listed in the table are recommended to study because they could cause potential clinical significant drug-drug interaction. And the EMA also recommended another two, it's the BSEP and the OCT1. And the PGP and the BCRP are widely distributed in many important organs and tissues such as the intestine, liver, and the blood-brain barrier. So FDA recommended all compounds to be tested to determine whether they are PGP or BCRP substrates. And depending on the clearance pathway, compounds also recommended to test if they are substrate of OATP 1B1 or OATP 1B3, or if they are substrate of OCT2, OAT1, and OAT3. Usually the cost of transporter substrate study is relatively high, so these assays are often performed in the later stage. PGP is the most extensively studied transporter. It can be studied using either KECO2 cell line or MBCK cell line in the presence and absence of verapamil. So here's our data show using KECO2 cell line with and without verapamil, and we find the PGP substrate co-choosing. There's a significant change when with and without verapamil. But propranolol, it is not a PGP substrate, has no change. So this is data in the KECO2 cell. And the same thing when we're using MDR1, MBCK cell line, MBCK2 cell line, with and without verapamil, we see that's a big difference for co-choosing, but there's no effect to propranolol. So for the inhibition, FDA and EMA recommended that all compounds needed to be tested to determine whether they are inhibitors of all those transporters. Those the same transporter is listed by both, recommended by both FDA and EMA. That is PGP, BCRP, OATP1B1, OATP1B3, OCT2, OAT1, and OAT3. And the EMA also recommended two extra, that is OCT1 and BSEP. And again, typically transporter inhibition assays are relatively expensive for earlier screening. So they often use the assay often performed in later stage. However, if using fluorescent substrates, the cost of transport inhibition assays can be substantially reduced. So if using fluorescent substrates, we can screen the transport inhibition in tier 1 screen in the early stage. Okay, now we move to metabolism. Metabolism is a major factor affecting compound stability and bioavailability. Metabolic stability is often the first characteristic to be screened. A paper published by Williams et al. In 2004 showed that metabolism is the major clearance mechanism for the top 200 prescription drugs in US in 2002. And the cytochrome P50 are the main enzymes responsible for the metabolism. So it is important to assess CYP stability as early as possible. Again, in our bioprint data show that this is from 476 matched data points compared bioavailability and metabolic stability. And it shows if a compound with a low metabolic stability usually it usually will not have a high bioavailability. Metabolic activity is described by enzyme kinetics. Enzyme kinetics parameters are calculated using Kinetics-Mentaine equation. And the assumption or assay condition we need make sure for the Kinetics-Mentaine study, it's first is the initial velocity, so that means the substrate consumption should less than 10%, and also the substrate concentration should be much greater than enzyme concentration, and also we need to make sure everything the time and the protein concentration is in the linear range. The problem for the using the Kinetics-Mentaine equation to calculate the intrinsic clearance in the early stage of drug discovery is this method requires using metabolites. However, in this stage, metabolites are often unknown. So then we have an alternative method that is using substrate depletion. The key parameter is intrinsic clearance will be calculated from in vitro half-life. For the accurate intrinsic clearance to be calculated, the substrate concentration should be much less than its Km. So typically, the substrate concentration or the test concentration for the test compound that we use is from 0.1 to 1 micromolar. And to minimize protein binding effect, small enzyme concentration is often used. Typically, it's from 0.1 to 0.3 milligram per ml liver microsome. So high intrinsic clearance may not be a good characteristic because it often results from a low Km. So compounds with low Km are easily saturated, resulting in nonlinear kinetics. So if we need to calculate Km in this stage without knowing what metabolite is, again, we still can use the substrate depletion method to do the study. In vitro intrinsic clearance can be used to predict hepatic clearance using well-studied model. High intrinsic clearance often suggests greater first pass effect and lower bioavailability. When we have metabolic stability intrinsic value, usually we will follow up with metabolite ID study and reaction phenotyping study. So now I will focus on reaction phenotyping. So the purpose of CYP phenotyping is to identify those isoforms that are responsible for the metabolism. If a CYP isoform that is responsible for metabolizing a compound, any drug inhibits its isoform will inhibit the metabolism of this drug and causing drug-drug interaction. So therefore, phenotyping is necessary for prediction of DPI. And also, if a compound is predominantly metabolized by polymorphically expressed CYP isoform, such as CYP2D6 and the compound may not be well metabolized by poor metabolized those individuals with mutated isoforms. And typically, the phenotyping is started using at least two of these three methods. The first method is using recombinant CYP isoforms to do the metabolic stability. The second method is using liver microsome, but with a specific inhibitor or antibody. And the third method is using a panel of single donor liver microsome for a correlation study. In the early stage, because metabolites are unknown, so using recombinant CYP isoforms may be the only reliable method. So here again, we will talk about CYP inhibition. CYP inhibition is the main cause of metabolic and metabolism-based DDI. And commonly two types of inhibition. One is reversible. The other one is irreversible. There are different modes of reversible inhibition. Some inhibitors compete with substrates for the active site, causing competitive inhibition. Some inhibitors bind to somewhere else and not compete with substrates for active site, but still affect enzyme activity. So those are non-competitive inhibition. So concentration of a probe substrate is key to get the useful result. The key kinetic parameter for enzyme inhibition is Ki. So using a substrate concentration equals to its Kn or much less than Kn, you can estimate Ki from IC50. Usually, IC50 value is more easier to obtain. Ki is a very useful parameter. It can be used to estimate whether inhibition could happen in vivo. So basically here it's based on the ratio of in vivo concentration to Ki. If it is greater than 1, inhibition will likely happen in vivo. If the ratio is less than 0.1, usually the inhibition will not likely to happen. And between 0.1 and 1 is probably. So we talked about the reversible inhibition. Now it's irreversible. So unlike reversible inhibition, irreversible inhibition often destroys enzyme itself. Therefore, even if the inhibitor is removed, the enzyme activity will not recover or very slowly recover. The inhibition often occurs when the enzyme attempts to metabolize the inhibitor. So that way, the longer the inhibitor incubates with the enzyme, the more potent the inhibition. So that's why it's also called time-dependent inhibition. A common protocol for study irreversible inhibition is pre-incubating the inhibitor or the test compound with the enzyme in the presence and absence of NADPH. With NADPH, the enzyme tries to metabolize the test compound, resulting in time-dependent inhibition if the compound is a time-dependent inhibitor. And after pre-incubation, the matrix is diluted tenfold, and the saturated concentration of probe substrate is added to analyze residual activity of the enzyme. The dilution and the high concentration of probe substrate is to minimize the potential reversible inhibition effects of the inhibitor. So here's the data for the CYP3A time-dependent inhibitor, troliandomycin. So here we can see, with pre-incubated with NADPH, it will have IC50. It's very potent inhibition, very low IC50. But without pre-incubated without NADPH, there's almost no inhibition. So now let's move to another very important DDI effect. It's CYP induction. So compound induced CYP activity are inducers. Induction mechanism is transcriptional activation. It's mediated through nuclear receptors. The major nuclear receptors that mediate the CYP induction are AHR for CYP1A, and the CAR for CYP2B6, and the PXR for CYP3A and CYP2C. The inducer binds to the nuclear receptor, causing gene expression, so mRNA level increase. And subsequently, CYP protein level also increase, and the resulting metabolizing activity increase. So CYP induction can be measured using enzyme activity or mRNA level as the endpoint. So in FDA 2006 guidance, enzyme activity was recommended for CYP induction. However, a study by Farmi et al. In 2010 indicated mRNA level is more sensitive than enzyme activity as the endpoint for CYP induction. So here we can see the sensitivity using mRNA is about 98. However, using enzyme activity is only 38%. So it's a big difference. Therefore, in FDA 2012 guidance, mRNA level was recommended as the endpoint for CYP induction. So here are the data using 10 known CYP inductions. 10 known CYP inducers and 5 known non-inducers. This is compounds selected from University of Washington drug interaction database. And from data you can see, apparently the individual difference is also quite large. Therefore, it is recommended to use at least 3 individual donors to conduct the CYP induction studies. So overall, in vitro metabolizing screen assay try to answer these basic questions regarding the investigated compound. First is the compound stable? And then which enzyme metabolize the compound? And also, whether the compound will inhibit or induce the major metabolizing enzyme. So based on this question, here is the recommended strategy for the primary screen. We can divide the primary screen to 2 categories. One is bioavailability. The other one is focused on the DDI, drug interaction. For the bioavailability, there's solubility, protein binding, permeability, metabolic stability. It's the important assay we need to run. And for the drug interaction, basically we will test if the drug is inhibitor of cytochrome 50 or transporters. And then based on the primary screening, we can have follow-up assays to study those more detailed or more expensive. Okay, so now I give time to Dr. Jack Michel.
Thank you very much, Jim. I'm going to move on to the toxicity screening, drug development pathway portion of the talk. And at this point in time, I would like to take the moment just to remind everybody of the goal of this webinar, and it's to characterize what type of early ADME and toxicity screening data is critical to move candidates further along a drug development pathway. I will largely be talking a little bit about cardiotoxicity, genotoxicity, and spending a bit more time on in vitro pharmacology screening. At this point, it's a good time to mention the idea of regulatory requirements. In this realm, they're a little vaguer than they might be in other places. And it's interesting to compare the idea of regulatory versus information that's just useful in making decisions. So what is clearly required by regulatory agencies are data that are very useful to have early on in the process to avoid any really bad surprises later on. I'll also mention, in at least a couple places here, the idea of a balancing act, that the more information you can have earlier in the process, the better, and that has to be balanced against the cost of generating that kind of information. But particularly in the case of these topics of cardiology, of cardiotoxicity, genotoxicity, and off-target pharmacological activity, the more information you have earlier, the easier it is to address when you're in the phase of lead selection or lead optimization, you can directly address these potential liabilities by making changes in the structures or choosing one lead over another, etc. When you get later on in the process, farther down, having already chosen your leads, then you have to manage those liabilities, either at the level of therapeutic levels, therapeutic index, metabolism, and those kind of things. So the earlier you can address problems, better. So these data are very good to have up front, and for that reason a lot of this testing can be done in a non-GLP regulatory mode, just to get the information you need early on. So the really clear cardiotoxicity indications are that you have to test for hERG, and there's hERG is a potassium channel that's involved in the repolarization after the cardiac action potential. Inhibiting this particular channel can cause that repolarization to happen more slowly, and thus will increase the QT interval, which can lead to cardiac fibrillations and sudden cardiac death, a pretty bad side effect. There's various ways early in the process to start addressing this. There's simple binding assays, which are probably your cheapest alternative. It's also your least reliable because you have a pretty good chance of having false negatives when you're assessing just one binding site on an ion channel. Better methods are automated patch clamp and manual patch clamp, and then in vivo studies farther down. And all of these things will have to be addressed at some point in the process, and the earlier you have this information, the earlier you can address potential problems. Moving quickly to genotoxicity, there's a couple different ways you can cause damage to the genome. They can directly damage the DNA itself, or you could interfere with the mitotic apparatus. There are a couple of types of tests that address this and are feasible to use fairly early on in the process. With respect to direct DNA damage, mutations and base pair substitutions, frame shifts, etc., can be detected using the AMES test. More or less subtle damage like double-strand breaks and interactions with mitosis can cause larger problems and cause fragments of whole chromosomes to form micronuclei in the cytoplasm, can be monitored using a micronuclease test, assay test, and I will give you a little bit of information on both these kinds of things. A lot of people will offer an AMES fluctuation test, which is a miniaturized version of the classic AMES plate assay. These tests measure the same thing you would measure in a classic AMES, which is the increase is the abilities to increase the mutation rate in the Salmonella bacteria. These tests are usually performed in 384 well plates rather than the agar plates, and thus require much less compound that get performed earlier in the process when the amount of compound available is not as great. There's also the potential to look at the possibility of metabolic activation, that is whether your compound, once metabolized, becomes mutagenic, and that's by incubating with an S9 liver fraction. And finally, good versions of this test will also include a bacterial cytotoxicity component, because compounds that kill the bacteria will look like false negatives. So the in vitro micronucleus test is a highly accepted test now. The one that we run is in CHO K1 cells, and it's run on an automated high-content assay system. This has been known as the Slomic system and other things, but basically it's a visual image-based system using plated cells. Once again, this is a test that uses a very low amount of drug, and it is a very highly accepted test for chromosomal aberration studies. In the figure you see on the right, this is an image that would be analyzed by the software. The cytoplasm of the cell is stained green. The nuclei, in this case binucleate cells, are blue. And there's a little yellow arrow pointing to a micronucleus, which is just a small amount of DNA extruded there from the nucleus. One looks through a bunch of these images and counts the occurrence of those micronuclei and looks for an increase in that number. So moving now to the in vitro pharmacology profiling part of that, there's a lot of different ways to approach the in vitro pharmacology screening. From the regulatory point of view, it's definitely required, but the ways people use pharmacology profiling is very different at different times in the process, and often people will have different screens at different times. Early on in the process, in the hit to lead and lead profiling, people run usually small profiles to characterize the chemotypes, their hits. They look for promiscuity screening. They look at selectivity and SAR. As they move farther into the process, the safety profiling aspect comes out with targets, using targets with known safety liabilities, usually human targets, and then benchmarking is also performed. That is for being able to compare a potential drug candidate to compounds that are already on the market, or competitors' compounds, or even other potential lead compounds. As you get farther on in the process to sort of the pre-IND phase, there's usually an expansion of the safety profiling, which means that we start profiling a much greater number of targets. A lot of these are safety targets. And the idea behind this is to have a more thorough de-risking of the potential safety targets. So, the advantages of early in vitro pharmacology profiling are, as I already mentioned, the ability to look at affinity determinations, structure and activity relationship studies, trying to determine promiscuity of chemotypes, and then as well as even the early detection of potential off-target activities that will end up being clinical liabilities, so that these things can be programmed out or designed out early on in the process. We think of pharmacology, in vitro pharmacology profiling, as being very cost-effective, and particularly when compared to in vivo work, in vivo models, because many compounds can be tested. You can test metabolites, which is important. You can test benchmark compounds. When it's done early in the process, very short turnaround times are used so that you can have iterative phases of design and going back and forth. Also, one other advantage is that once you've run a profile, there are follow-on assays to determine the functional consequences, whether it's an agonist or antagonist at a receptor, or in a number of different in vitro and vivo models. You can follow up those hits in a fairly logical fashion. And as mentioned before, it allows for the side-by-side comparison of lead compounds. Another way to do the comparison is that these profiles allow more bioinformatic approaches to predicting the potential in vivo effects of those compounds, looking at profile similarity of your compound with other compounds you've run before, or with drugs that are on the markets. So just to bring back the idea of the balancing act and to re-summarize what I've been saying, I created this slide here, which is the timeline going from left to right of hit to lead all the way to pre-IND. I've created two very beautiful wedges here. There's the brown assays wedge and the number of compounds wedge in gray below that. And early on in the process, in the hit to lead process, we often have very small panels of assays, small numbers of assays, and large numbers of compounds. And for these type of panels, like I said, we've discussed selectivity and structure-activity relationships and promiscuity. Towards the middle of the process, when you start looking more into safety liabilities, you have a greater number of assays, but fewer compounds are passing between those. And finally, as you've chosen your lead and you're really trying to have a real thorough understanding of what the potential problems are, you can really start adding a much larger panel of assays and explore a greater number of targets. Some people run as many as 200 to 300 different targets there, just to make sure that they have a really clear idea and to avoid any potential surprises farther down. But by the time they're doing that, they're only running the panel on one or two compounds. It's kind of a good time to mention that fairly recently, in order in some way to fill a little bit the vacuum that has come from the regulatory agencies with respect to what should be run and when, some groups from AstraZeneca, GlaxoSmithKline, Novartis, and Pfizer got together to combine their joint knowledge and their joint experience in doing safety pharmacology profiling. And the idea was to come up with a panel of assays, primarily oriented towards safety that could be run, that made sense, and that were very much information rich. And it was a very nice piece of work done by all these people, and they came up with a panel of about 44 different targets, each of which was closely linked to fairly well-known drug liabilities. We, as a company, offer this panel. We've also expanded that by about twofold with other targets that could be added on later that we also believe are fairly closely linked to fairly well understood liabilities. The next slide here is just a little selection of those assays, but in general, each assay can be linked to specific adverse drug events, and usually the more serious ones, at least at this phase of the work. What's important to know about is that pharmacology screening is not an end in and of itself. Follow-up has to be done, but these are important data. In this table here I've constructed, I've listed several targets, the ADRs they're associated with, and some drugs that, at least in red, have had to be withdrawn from the market due to those ADRs. So for instance, hERG inhibition, causing those ventricular arrhythmias we spoke about, trifenadine and stemazole, cisapride have had to be removed from the market. Next line down is 5-HT2B serotonergic receptor, in which agonists have been shown to be related to cardiac valvulopathies, and that fenfluramine and pergolide have had to be removed from the market. But the main point of this slide is that these targets can be linked to very serious side effects, and that there is follow-up work that has to be done. There are in vitro and in vivo models that have to be followed up once you've decided you have a particular hit on any of these targets. Just to talk a little bit about the value of benchmarking, as I've mentioned it a couple times already. Right here I'm showing you a slide, heat map of the pharmacological profiles of a series of antidepressant drugs. They've been divided into three groups. The drugs themselves are on the vertical axis, and the targets are on the horizontal axis. As this is a heat map, red shows a strong hit and white shows a little or no hit. And these have been grouped somewhat historically. This is sort of the medicinal history of antidepressant drugs at some level. And the first group of drugs are largely tricyclic antidepressants, the first generation of antidepressants. And you can see that their primary activity, or what's believed to be their primary activity, may not necessarily be true, is the 5-HT transporter and inhibition of that. But they have a lot of other profound activities across there. Those are very rich profiles with strong hits across a lot of targets that can be attributed to a lot of the fairly severe side effects of these compounds. These drugs were followed up with a second generation of very, very successful drugs, paroxetine, sertraline, and fluoxetine. And you can see that here in this comparison, while they all retain the same 5-HT transporter activity, their off-target activities are much, much less. The final block of compounds is our third and fourth generation antidepressants, and they have even cleaner profiles. But when you look at a slide like this, you can see how that any group trying to develop a new antidepressant would certainly like to see where their compound fell with respect to this history of this particular therapeutic area. And this kind of work can be done across any number of different therapeutic areas. So having given you a general framework on how in vitro pharmacology is used throughout the drug discovery process, I thought I would show you a little bit more of some of the intricacies. Starting with a slide that I titled, "It's Not That Simple." And what I'm going to do here is just go through the 4 topics here that show you some of the finer points of in vitro pharmacology profiling, some of the problems and things you need to take into account. I'll talk about prodrugs and metabolites, species differences, antagonist versus agonist radioligands for binding assays, as well as the question of how well binding assay data correlates to functional assay data. And for each of these, I'll just show a single slide and try and give you a little bit of a taste of some of the finer points. So in this slide here, I'm showing you another heat map, in this case, of a drug that was withdrawn from the market, terfenadine, and the drug metabolite, the primary metabolite of that compound, which has since replaced it. And what you can see in the heat map there, the top compound is terfenadine. That's on the vertical axis. Below that is fexofenadine, and there is a fairly large panel of in vitro pharmacology assays there. Once again, red is a strong hit, and green is going to be no hit. And what we see is two compounds with very, very similar structures, yet very, very different pharmacological profiles. And usually people operate on the idea that compounds with similar structures have similar profiles, and that holds true a lot of the time, except when it does not. And this is the case where it does not. So looking at this fairly large difference between compound and its metabolite can really show you the interest in running parent compounds and prodrugs and their metabolites through these kind of panels of in vitro pharmacological assays. Another thing that people have to account for is species differences. In this case, I'm showing you two different versions of the histamine H1 G protein-coupled receptor binding assay. Here we're looking at a plot of a rabbit version of the histamine H1 and the human version, and we're looking at percent inhibition. And if these two receptors, one in each species, correlated perfectly, you would see a very nice line going from 0 out from the 45-degree angle. But you do not see that in this graph here. You see that there's some pretty big differences between the rabbit histamine H1 and the human histamine H1. Other receptors across species line up much better than this, but this is a case where it doesn't. And these things often need to be taken into account when you're thinking about what animal models might be doing for toxicology testing down the road. Another thing we run into is that historically binding assays have been around for a very long time, and there's a lot of history associated with them and the way they were developed, and there are very classical radioligands that have been used. Historically, it's been mostly antagonist radioligands, but we do find that there are some differences. In this case, I've plotted a dopamine D2S assay using an antagonist radioligand on the horizontal axis, and on the vertical axis we have the same target but using an agonist radioligand. And once again, you do not see very good correlation. And we know that for some of the classical antagonist ligands that have been used for many years, even the endogenous agonists are barely hits on these kind of targets. So that's another aspect to take into account in selecting what assays you want to use. And finally, a question we often get asked because, as I mentioned before, a lot of the in vitro pharmacology bindings need to be followed up if they're hits. And so people generally want to know whether there's a good correlation between the binding assay data and the functional assay data. And after having looked at quite a few of these, generally the way we approach this kind of work is to use the same cell lines for the binding assays that we use for the functional assays. So we've had a chance to look at a lot of these cell lines, we believe pretty much in the same background. But we do find that the correlation is sometimes difficult to establish between the binding and the functional. In the case of this particular assay, the 5-HT2B binding assay, which is a particular interest for the cardiac valvulopathy we mentioned earlier, we found that in this case we're looking at the binding assay on the horizontal axis and the functional assay on the vertical axis, and we're looking at PIC50, so it's the inverse log of the IC50. And what we do see in the case of this particular receptor is that when we're looking at antagonists, we get a relatively good correlation between the binding and the antagonist effect. However, when we're looking at agonists on the right, that correlation somewhat disappears. And this is not necessarily a general effect. It really is a characteristic of the assays, and it's definitely something you need to take into account. So finally, just in conclusion here, I've gone through a bit of what the requirements might be for in vitro tox and say those regulatory requirements, and they are in fact quite a bit vague. Things we do know is that you really have to address cardiotoxicity, and most specifically hERG, and we discussed ways we could do that. Genotoxicity needs to be addressed in the form of AMES and micronucleus type assays, and in both cases, the earlier you have information on these, the better, because they will have to be addressed in one fashion or another. In vitro pharmacology needs to be performed. However, no specific requirements are really delineated within the guidances and the regulation and the regulatory documents from the regulatory agencies. As I told you before, that consortium of pharma got together and tried to put forth a list that they thought was reasonable, and it's gaining some traction and may be useful for that. Once again, this is best addressed as early on in the process as possible. If you have strong off-target activities and you detect them at the level of leads or lead selection, you could choose a better candidate that has less off-target activities, or you can send it back to the medicinal chemist to try and design out some of those potential liabilities. Comments from inside the agency, while they don't say something specific, they say they like to see a sufficient diversity of data, or sufficient diversity of targets and data, and more importantly, you can't just run the assays and send them the data. You need to follow up. If you have hits, you have to account for them in one fashion or another. You need to justify that there is not much risk associated or not, and that's very important. Just generating the data is not sufficient. So that's pretty much all we have for today. We'll go to the questions in a second, if there have been some posted. Okay, yeah, so I see. So one question we got was the correlation of binding to functional assays. Was it more for agonists, antagonists, MS for agonists? Is this a general phenomena? I can't say it is always that way. It seems that there is a certain amount of that happening, and that the antagonists do tend to have a better correlation than the agonists, but that's not always the case. I wouldn't want to completely generalize. So another question we have here is, instead of using hERG, do you recommend to check cardiotoxicity with cardiac myocytes? Also, which advantages and drawbacks do you see? So I would disagree with the instead of using hERG part, because you have no choice. You have to address that directly. But doing a cardiac myocyte type of experiment is very informative and will provide a lot more information, much more complete picture of the cardiac toxicity than just running hERG. But you will have to individually address hERG in any case.
All right, thank you for those answers and the presentation. We've now reached the end of the question and answer portion of the webinar. If you do have further questions, please direct them to Dr. Tang's or Dr. Majone's email address shown on your screen. We will compile a list of more questions with your answers, and we'll send that out to everyone. Thank you for participating in today's webinar, and you will be receiving a follow-up email from Eurofins with access to the recorded archive for this event. Also, as you're leaving, a survey window will be popping up on the screen. Your participation is appreciated because it will help us improve our future webinars. So now please join us in thanking our speakers, Dr. Tang and Dr. Majone. Thank you. We hope you found this conference informative. Please have a great day. Thank you for attending.
Good job.
Thank you.